It starts with a bone smaller than a fingernail. In the summer of 2008, a team of Russian archaeologists was working through the sediment of a cave in southern Siberia, a place so remote that the nearest town is hours away by dirt road. They had been digging in this cave for decades. They knew its layers. They knew its stone tools, its animal bones, its ash pits from ancient fires. What they did not expect, buried inside a layer of hardened sediment, was a single fragment of a finger bone, so small it could sit on the tip of a pen. At first, nobody thought much of it. Cave floors are full of bone fragments, animal, human, ambiguous. This one was bagged, logged, and set aside with the rest. 2 years later, that tiny sliver of bone would rewrite the story of who else was walking the earth alongside our ancestors. It would introduce the world to a kind of human science didn't know existed. And it would do it not through a skull, not through a skeleton, but through a chip of finger bone and a strand of DNA too degraded for anyone to have predicted what it held. This is the story of Denisova Cave, and the discovery that started with almost nothing and ended up changing how we understand human origins. The cave sits in the Altai Mountains, a range where Russia, Mongolia, China, and Kazakhstan nearly touch. Locally, it's named after a hermit called Denis, who is said to have lived there in the 18th century. But long before Denis, and long before any human alive today, this cave had already hosted visitors for hundreds of thousands of years. Archaeologists had worked the site since the 1970s. It has 3 interconnected chambers, roughly the footprint of a modern house, with a wide entrance that lets in enough daylight to make the main chamber usable without torches. That's part of why it was occupied again and again across an almost unimaginable stretch of time. Layer after layer of sediment, stacked over hundreds of thousands of years, had preserved stone tools, animal remains, and, every so often, fragments of the people who passed through. By the time the fingerbone fragment was found in 2008, the site had already produced Neanderthal remains. That wasn't a surprise. Neanderthals are well documented across us. Eurasia and Siberia was within their known range. So when the bone was sent off for analysis, the expectation was ordinary. Confirm it was Neanderthal, log it, move on to the next question. The sample went to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, to a team led by geneticist Svante Pääbo, a scientist who had spent years pioneering methods for extracting usable genetic material out of fossils that were, in most labs, considered too old and too degraded to bother with. Pääbo's team had recently made global headlines for sequencing the Neanderthal genome. The Siberian fingerbone looked on paper like it would be a footnote to that larger project. It wasn't. To understand why this particular cave kept producing results other sites couldn't, it helps to know what makes a site good for preserving DNA in the first place. Ancient genetic material breaks down over time, and heat speeds the process along. A cave in a tropical climate might destroy usable DNA within a few thousand years. But Denisova Cave sits at altitude, in a region with a cold, stable climate, and its sediment stays consistently cool year-round. That combination, cold, dry, undisturbed, is close to ideal for keeping fragile genetic material intact for tens of thousands of years. It's part of why a single cave in the Altai Mountains has produced more usable ancient hominin DNA than almost anywhere else on the planet. Before we get to what the DNA revealed, it's worth pausing on why this method works at all, because it's the key to the entire story. A bone doesn't just tell you its shape. Buried inside its cells is genetic material, fragile, and over tens of thousands of years, badly broken down into short, damaged pieces. For most of the history of paleontology, that damage made ancient DNA effectively unreadable. Pääbo's lab had developed techniques to extract, sequence, and reassemble those broken strands, matching overlapping fragments the way you might rebuild a shredded letter, one scrap at a time. It's slow, it's exacting, and by 2010 it was finally reliable enough to apply to a bone this small and this old. When the team sequenced the mitochondrial DNA from the Siberian fingerbone, they compared it against 2 known references, modern humans and Neanderthals. That's normally where the comparison ends. You match to one or the other and you're done. This time, the sequence matched neither. The differences between this DNA and modern human DNA were roughly twice as large as the differences between Neanderthal DNA and modern human DNA. In genetic terms, that's a significant gap. It meant the individual this bone belonged to wasn't a Neanderthal wearing an unusual genetic signature. It was something else. A lineage that had split off from the ancestors of Neanderthals and modern humans, gone its own way, never been identified before. The researchers published their findings in December 2010 in the journal Nature. The bone belonged, they proposed, to a distinct group of archaic humans, later named Denisovans, after the cave where the fragment was found. A tooth recovered from the same cave, examined separately, turned out to belong to the same population, distinct in shape from anything seen in a Neanderthal or a modern human jaw. The tooth was notably large, roughly double the surface area of an average modern human molar, which told researchers something physical about this population even before a single complete skeleton had ever been found. Two pieces of physical evidence, one entirely new branch on the human family tree. For a field that usually measures progress in single-digit fossil finds spread across decades, this was seismic. A previously unknown group of humans, identified almost entirely through genetics rather than shape or skeleton, was something that had never happened before. Denisovans became the first hominin species ever described primarily through DNA, and that raised an obvious question. Who exactly was this person? Later, analysis narrowed it down. The bone came from a female, likely between 5 and 7 years old at the time of her death, young enough that the growth plate in the bone hadn't yet fused, which is how researchers estimated her age. She's been referred to since as Denisova III, though informally, some called her the X-woman, a nod to how little was initially known about her beyond the X, marking an unidentified variable. Dating a bone this size, in a cave with this complicated a history, turned out to be its own separate challenge. And it's worth being honest about that difficulty because it's part of what makes this story credible rather than tidy. Early estimates placed the fragment somewhere between 30,000 and 50,000 years old, based on the layer it came from and comparative dating of the surrounding sediment. Later studies, using more advanced Bayesian modeling that combined radiocarbon dating, sediment analysis, and genetic mutation rates, pushed some estimates further back, suggesting she may have lived at least 50,000 years ago, with a wider range extending toward 76,000. Different methods applied to a bone chip this fragile don't always converge on a single number, and archaeologists have been upfront about that uncertainty rather than papering over it. What's consistent across the estimates, though, is the general window, tens of thousands of years ago, deep enough in time that this cave and its inhabitants predate almost every written record humanity has ever produced. Once Denisovans were confirmed to exist, the second question became unavoidable. Were they alone in that cave? The answer, it turned out, was no. Neanderthal remains had already been found at Denisova Cave. Now, genetic surveys of the sediment itself, not just bones, but the DNA embedded in the cave floor, showed that Neanderthals and Denisovans had both been present, sometimes within overlapping windows of time. That meant two distinct human lineages, evolved separately for hundreds of thousands of years, were passing through the exact same cave system. That leads to the detail that turned this from a fascinating discovery into a genuinely stunning one. In 2012, researchers recovered a long bone fragment from the same cave, one piece, among more than 2,000 unidentified bone scraps collected over years of excavation. For years, it sat uncatalogued, indistinguishable from animal bone by eye alone. It wasn't until 2016 when a team applied a method called collagen fingerprinting, essentially reading the protein signature of a bone fragment to determine whether it's human before even attempting DNA extraction, that this particular scrap was flagged as worth a closer look. When the DNA came back, it showed something no one had ever confirmed before in the fossil record. This individual's mother was Neanderthal. Her father was Denisovan. She wasn't just evidence that these two groups had crossed paths. She was living proof in her own genome that they had interbred. A first-generation hybrid, a daughter of two different kinds of ancient human. Researchers nicknamed her Denny. She's estimated to have lived roughly 90,000 years ago, and to this day, she remains the only first-generation hybrid hominin fossil ever identified. Sit with that for a second. Out of the millions of bone fragments excavated from ancient sites around the world, exactly one has ever been confirmed as belonging to a child of two different human species, caught in the act, so to speak, of two ancient lineages meeting. The implications didn't stop with the cave itself. Once scientists had a reference genome for Denisovans, they could search for traces of that DNA in living people today. And they found it, not in Siberia, not in Europe, but scattered through the genomes of modern populations across Asia, and most strikingly in people native to Melanesia and parts of Oceania, where Denisovan ancestry can make up several percent of an individual's total genome. That's a meaningful fraction, high enough that some of it appears to have shaped modern biology in measurable ways, including genetic variants linked to high-altitude adaptation found in Tibetan populations, which some researchers have traced back to a Denisovan origin gene passed down over tens of thousands of years. In other words, an extinct population identified from a finger bone the size of a pebble had, in some quiet biological sense, never fully disappeared. Pieces of them are still here, embedded in the DNA of people alive right now. This is usually where a story like this would wrap up. New species discovered, mystery solved, headline delivered. But Denisova Cave kept going. Excavations continued through the following decade, and the picture kept growing more complicated in the best possible way. Researchers identified bone points and pierced animal tooth pendants in the cave, artifacts associated with more advanced tool use and personal ornamentation, dated to roughly 43,000 to 49,000 years ago. Whether Denisovans or early modern humans made those particular objects is still an open question. The evidence doesn't cleanly assign credit to one group or the other, and honest researchers say so rather than guessing. Then, in 2022, came another jolt. A team led by archaeological scientist Katerina Duca went back to the oldest, least explored layers of the cave, sediment nobody had successfully pulled hominin fossils from before, and found 3 more Denisovan bone fragments. These weren't from the same time period as the famous finger bone at all. Dating placed them at roughly 200,000 years old, nearly 4 times older than the original specimen that started the story. For the first time, researchers were also able to link Denisovan remains directly to specific stone tools found in the same layer, giving a clearer picture of how this population actually lived, not just that they existed. Put the layers together, and Denisova Cave stops looking like the site of a single discovery and starts looking like a multi-100,000-year record of at least 3 human lineages, Denisovans, Neanderthals, and eventually modern humans, moving in and out of the same shelter, sometimes generations apart, sometimes close enough in time to meet, and at least once, close enough to have a child together. It's worth being clear about what's firmly established here and what's still being worked out, because that's the honest shape of this kind of science. What's established, Denisovans, are a genetically distinct hominin population, confirmed through multiple independent fossil finds and DNA analyses, first identified in 2010 and expanded through discoveries as recent as 2022. What's established? They interbred with both Neanderthals and early modern humans, and that ancestry persists in people alive today. What's still unresolved? Their exact physical appearance? No. Complete skull or skeleton has ever been found, so reconstructions rely heavily on inference from a jawbone, a handful of teeth, and fragments of bone. What's still debated? The precise age range of individual specimens, given how difficult it is to date material this fragile with total precision, and exactly how widely Denisovans spread across Asia before disappearing. That last point connects to a genuinely strange piece of the puzzle. For years after the initial discovery, every confirmed Denisovan fossil came from this one Siberian cave, nowhere else on Earth. Then, in 2019, a mandible discovered decades earlier on the Tibetan Plateau, in a cave more than 2,000 kilometers away, was re-examined and confirmed as Denisovan through protein analysis, dated to at least 160,000 years old. It was the first physical proof that this population had ranged far beyond the Altai Mountains, matching what geneticists had already suspected from studying modern DNA. A molar found in Laos, reported in 2022, may extend that range even further into Southeast Asia, though researchers have been careful to call it evidence still requiring confirmation rather than settled fact. So the population that the world first met through a finger bone the size of a grain of rice turns out to have spread across a huge stretch of the ancient world, from icy Siberian mountains to the high plateau of Tibet, possibly as far as the tropical forests of Southeast Asia, surviving for at least 200,000 years, overlapping with 2 other human lineages, and leaving behind a genetic legacy that quietly persists in millions of people today. In 2022, Svante Pääbo, the scientist whose lab first sequenced that original finger bone, back in 2010, was awarded the Nobel Prize in Physiology or Medicine for his work on ancient genomes and human evolution. It's a rare thing for a Nobel Prize to trace directly back to a single fragment of bone pulled out of cave sediment by hand, but that's essentially what happened here. A find so small it almost didn't get a second look ended up reshaping how an entire field understands the deep human past. What makes Denisova Cave remarkable isn't just that scientists found something unexpected. It's that they found it through the smallest, least promising piece of evidence imaginable, a chip of bone that, by every normal standard, should have told them nothing. Instead, it told them that human history is not a single line running cleanly from ancient ancestor to modern human. It's a tangle. Different populations evolving in parallel, crossing paths in caves like this one, sometimes leaving nothing behind but a name we invented for them centuries later. The cave is still being excavated. Every new field season carries the possibility of another fragment, another layer, another population we haven't identified yet, quietly waiting in sediment that's been undisturbed for 100,000 years or more. So here's a question worth sitting with. If a species-defining discovery came from a bone, fragment the size of a fingertip, how much of our own history is still buried, unnoticed, in places we've already dug? If you had to guess, how many more human lineages do you think are still waiting to be found in caves like this one? Drop your guesses in the comments. And if this kind of deep time mystery is your thing, there's more of it waiting for you right here.